with that, I would like to hand over directly to the next speaker, who is fortunately as passionate about pigs as Dr. Clayton Johnson. His name is Dr. Oliver Duran, and this one from all the three speakers I probably know best because he's also my boss. And therefore, I will only use kind words for the introduction. But let me tell you that from my perspective, there are two reasons why Dr. Oliver Duran is a really perfect match for this presentation and for this event. Because at the one hand side, he's the leader of the global technical swine team, and he is really incredible knowledge when it comes to swine disease and management. And Oliver told me that back then, as he was a swine practitioner in the US, he had nightmares about PRRS and abortion storms. So these two features really make him a specialized speaker for this dedicated PRRS event. And in the next 20 minutes, Dr. Oliver Duran will explain about PRRS control of type 2 strains in mixed type 1 and type 2 infections. Oliver, the floor is yours.
Thank you, Myers, for your kind words, and I'll keep those in consideration when we do your annual review. Well, good evening and good morning, everyone. It's a pleasure to be here with you today. You know, perhaps a bit of a contrast with Clayton's presentation. I will try to share with you what the science says regarding type 1 and type 2 PRRS and what the emergence of type 1 in Asia means for cross protection. Hopefully, after these 20 minutes, we'll be able to make some nice conclusions. The order of the topics that I'll speak about today is here. First, I'll share with you a little bit the published data on prevalence of PRRS type 1 and type 2 in Asia. Then talk a little bit about how these viruses behave on farm or in challenge studies in terms of their pathogenicity. Then we'll talk a little bit about the control tools that we have. As Clayton Johnson mentioned, we have vaccines as one of our main tools to control PRRS, let you know how those vaccines do against these heterologous challenges. And then a very quick overview really about how we should approach control. So first, just really short introduction on this virus. I think all of you unfortunately know it very well. And I do still have nightmares about this virus. I think it's important to understand some very basic concepts. One is that there is now very clear distinction between two types within PRRS. Type 1, also called European or EuroPRRS or PRRS1, and then the PRRS type 2 or North American type virus. Both of them are arteriviruses. Both have very similar characteristics in terms of their genetic makeup. They have 10 open reading frames. And it is important that we understand that at the genetic level, they only share about 50 to 60% genetic identity. So these viruses, while they're both in the same family, they are quite different. The other thing that you know already is that these viruses, this population of viruses is changing all the time. And so we are seeing new emerging strains that are challenging our production, are challenging our control efforts. This map gives you a very quick overview of where PRRS is in the world. The gray areas, there's not many of those, are free from PRRS. And the others are divided between type 1, which is the predominant strain in Europe, and then the type 2 PRRS strains, which are the predominant strains in the Americas and in Asia. It's also clear, and you'll see this in a minute, that the predominant type circulating in Asia is type 2, although it's fair to say that this picture is not as clean as it was perhaps 10, 15 years ago. So both in North America and in Asia, type 1 strains are being detected. And we also detect in some countries type 2 strains in Europe, although much less frequently. So let's start with China. Reviewing the literature, it's clear that type 1 strains are circulating in China, have been circulating in China, but really they are detected and sequenced relatively infrequently. And while there are some reports of positive identification of PRRS type 1 in China, the detection rate is still low. And most of those strains that are found are within subtype 1, which is the most prevalent type within Europe. And maybe it came to China through breeding stock or other means. This chart, this paper from Jiang et al. Just last year, gives you a very nice illustration of kind of how PRRS has changed, kind of a snapshot of the evolution of PRRS within China. And you can see that even for type 2, there has been a number of predominant strains that have then faded away while new strains have emerged, like the NAD30-like strains, for example. What you can see from this too is that while EU or type 1 strains are detected, it's relatively infrequent. In another study, a couple of studies published in the last 2 years, it's clear again that type 2 strains are the predominant strains circulating in China. And for example, you can see from Chen et al. In 2020 that out of 712 clinical samples collected in the whole of China during this period, PRRS was confirmed in 100 samples, of which only 2 were PRRS 1 or PRRS type 1. So again, it's present, but at a low level. Let's look at the picture in other parts of Asia. If we look at Thailand and Southeast Asia, a paper published a few years ago now, and maybe the situation is evolving, and this is all that we could find in the literature, is that really in this part of Asia, type 2 is clearly the most frequent detected strain, although type 1 has been identified in Thailand. But in other countries in Southeast Asia, it appears that type 2 is very much predominant, even though we know that type 1s have been identified in Malaysia, Indonesia, and other parts of Southeast Asia. Vietnam, again, some of the surveys have been done a few years ago, but those did not recognize PRRS type 1 in those surveys. So in Vietnam, at least in the literature, it appears that type 2 is predominant, and to date, type 1 has not been described in the published literature. Where things change a little bit is in Korea. So Korea, unlike other Asian countries, has a pretty widespread distribution of PRRS type 1 isolates across most of the provinces within Korea. A study published 3 years ago analyzed 631 PRRS positive samples and sequenced OR5 from these, and they confirmed this distribution. So both type 1 and type 2 were present, but also, in addition, there was herds or samples that had both types detected. The authors also describe a particular population of strains that are very similar to commercial type 1 PRRS vaccines. And they also describe that NADC30 is NAD30-like viruses are also spreading in Korea. It's also important to understand that some of these vaccine-related isolates, particularly those associated with the Unistrain product, do appear to be associated with clinical outbreaks on farms. And I share with you two papers that describe this recently. And so this is a cause for concern, and you know, this brings particularly a lot of our academic colleagues into questioning the use of MLVs. And I think it's very important that we keep an eye on this situation. But again, it's important to understand all of the background of these two papers, but it's a risk that we need to understand. Okay, so PRRS type 1 is present in Asia, but is much less frequently detected and sequenced on most farms within Asia. The second point I want to make is that when you review the scientific literature, it's clear that PRRS type 2 is more pathogenic than PRRS type 1. Now, this is a broad generalization, and with PRRS, you have to be very careful to make generalizations because there is a multitude of different strains with different pathogenicity, strains that behave very severely in field cases. When you then put them into pigs in the laboratory, they do not express severe pathogenic picture. So it's quite difficult to assess this question. But what I share with you here is a list of reasons or findings that support that in general, in the majority of cases, PRRS type 2 is a much more severe enemy or adversary than type 1. These are some of the parameters. Based on a very large study that Mora and colleagues at Iowa State carried out, looking at different batches covering around 55,000 pigs, they found that exposure to type 1 was not associated with higher mortality, while those batches that had confirmed PRRS type 2 exposure were suffering from higher mortality. When we look at clinical respiratory disease, challenges have been done in controlled conditions, comparing type 1 versus type 2 strains, and clearly type 1 strains in these challenge models did not result in clinical disease, while type 2 did. When you look at lung lesions, type 2 viruses cause more severe lung lesions in side-by-side comparisons, and this is pretty frequently shown in the literature. If you look here from the very early studies done in 1995, all the way to studies published last year, show more severe lung lesions for type 2 viruses. When we look at rectal temperature, it's also clear that type 2 viruses cause higher rectal temperatures in these challenge studies. If we look at presence of PRRS virus in lungs, here there are less studies, but it's also being confirmed. We can see that the replication rate for PRRS type 2 is higher, both in studies done in Korea and elsewhere. And we also find that when we look at field outbreaks, the duration of viremia is longer and higher in PRRS type 2 infected populations. And finally, two more points. When we measure PRRS through detection of the virus in air, by air collection, this has been very, very difficult to do with type 1, but it's relatively successful and easy to do with type 2 viruses. So airborne clouds of virus seem to be more likely with PRRS type 2. And finally, in some studies, when looking at the ELISA, the IgG ELISA response, it seems like pigs exposed to type 2 PRRS show a higher and earlier IgG response. So let's just illustrate a couple of these points. This is data from challenge studies where they looked at 3 strains, 3 type 1 and 3 type 2 strains, and measured rectal temperature daily during this experiment. And what you can see visually is that the 3 type 2 strains resulted in fevers and average temperatures that were higher than the type 1 challenge situations. If we look at a field infection, this is a case published recently in Russia where they had both type 1 and type 2 infection circulating at the same time in this large herd. What they detected here is that the exposure to PRRS affected more of the sample population, and the positive population remained positive longer, as you can see from this graph. So I think one way to illustrate this to you is that while both of these products are alcoholic drinks, if you've had a bottle or maybe half a bottle of Baijiu compared to a bottle of beer, the next morning you will feel very differently. And I think in a way this is a humorous way of explaining how these 2 viruses behave in pigs. It is also important to share with you that pathogenesis is difficult to compare, and sometimes in some studies it has not been possible to differentiate or to say that one is more pathogenic than the other. And this is a lot to do with genetic variability, both within each of the type 1 or the type 2, but even between them. So the analysis is always going to have that question mark, if I test 2 strains, do they represent what I have in the field? It's also very important to understand that what we see in a lab challenge and what we see in the field may look very, very different with the same isolate. And this can be very frustrating to really try and understand what's going on with these PRRS strains. The other thing it's important to tell you is that if you have a naive pregnant sow, it doesn't matter if you challenge her with a type 1 or a type 2 virus, there will be consequences to that fetus. So in this very sensitive part of the gestation, of the pregnancy, you will get clinical disease, you will get infected fetuses, you will get stillborn pigs, abortions possibly, because this is the most sensitive model we have for PRRS. The other thing that is quite important is that in some studies, there was limited differences between type 1 and type 2. So some strains of type 1 are certainly pathogenic and can cause pretty severe clinical disease. The other thing that is becoming clear is that in herds that are, or in situations where we have co-infection of type 1 and type 2, we can have pretty severe clinical pictures, either in breeding animals or in piglets. So once again, what this tells me, one of the conclusions that I make from all of this is that, biosecurity has always been important, but we need to understand that if we keep bringing new strains, whether it's type 1 or type 2, into our herd, they will behave like a new infection and will cause severe clinical disease. So biosecurity, biocontainment is more important than ever, as you can imagine. There is some evidence in the literature that type 1 modified live virus vaccines do not work against type 2. And I share some of those references with you here. These are all peer-reviewed papers. What they show is that while there may be some degree of protection against the type 1 challenge, in these experiments, the type 2 challenge strains often resulted in overcoming the immunity of the vaccine. And there's examples in pregnant gilts, like Yong et al. In 2017. There's a study also from Korea that showed that type 1 PRRS-MLV vaccines protected against the type 1 challenge, but provided no protection against heterologous type 2. In a mixed infection, this was carried out by researchers in Thailand and published just this year, Madapong et al. It showed that type 1 PRRS-MLV was not able to reduce lung lesions when compared to unvaccinated controls when challenged with the type 2 virus. Field studies have also been carried out, and type 1 vaccine was not able in this dual infection to reduce lung lesions or improve the average daily weight gain. And that was published by Yang et al. In 2020. And finally, in boars, we know that to achieve good protection in boars, you probably need a high degree of homology to reduce viremia or semen shedding. And so in my opinion, we shouldn't be vaccinating boars in boar studs. Boar studs should be PRRS naive, producing negative boar semen. But vaccine was not able, type 1 vaccine was not able to prevent shedding in semen of PRRS type 2 challenge virus. In dual infections, vaccination with type 1 PRRS-MLV was not able to control this dual infection. It was able to reduce the viremia levels for type 1 in this challenge model, but not type 2. And it was the same with lung lesions. The type 2, the pigs vaccinated with a type 2 vaccine did have a reduction of lung lesions after challenge, but not the modified live type 1 vaccine. And the authors explain that this may be related to the inducing of interferon alpha, specific interferon alpha secreting cells that seem to be produced at a higher level with type 2 PRRS-MLV. So what about the other way around? What about evidence in the literature that type 2 based PRRS-MLV vaccine protects against a type 1 challenge, and therefore cross protection across genotypes now? And there are some papers in the literature that support a degree of cross protection. Again, in none of the cases will the protection be complete. We know that's the case with even type 2 strains, that we don't completely induce sterilizing immunity with modified live virus vaccines. But this evidence shows that type 2 PRRS-MLV provided protection against a type 1 challenge, whether it was in a late-term pregnant gilt model, like Yong et al. In 2018, or when looking at piglets and measuring the CMI response, type 2 based MLV vaccines were able to produce a powerful cell-mediated immune response against type 1 challenge strains. And finally, there's data from a Korean study that vaccination against type 1 challenge was effective with the type 1 vaccine, but really only PRRS type 2 MLV vaccines protected against both these Korean type 1 and type 2 strains in a challenge study. So quite a bit of evidence of cross protection for type 2 based MLV vaccines. So what about Engelbach PRRS-MLV now? Let's have a look at what's in the literature for the vaccine that Boehringer Ingelheim produces. Well, there is evidence in the literature that I'm going to share with you. First, some evidence, some recent evidence from Asia. These are two papers published at the APVS in Busan. If you remember, we had meetings where we could actually meet. In this two papers, one was a challenge study with type 1 and type 2 PRRS, where Engelbach PRRS-MLV demonstrated reduced lung lesions, reduced PRRS detected in lungs by staining, and also reduced virus detected in blood. The second is a case published by veterinarians in Malaysia. And this is a case report, a field report from a farm that was infected, naturally infected with both a type 2 and a type 1 virus. In this herd, the vaccination program was with a type 1 based vaccine, and they encountered a very severe outbreak. And when the vaccination in this herd was switched to Engelbach PRRS MLV, there was a very dramatic improvement in clinical and productivity results on this farm. There was two important factors, in my opinion, here. One was the use of a type 2 vaccine, but perhaps as importantly, it was a whole herd vaccination. So both the breeding herd were mass vaccinated twice at the beginning of the outbreak, and then the piglets were vaccinated to ensure that we really shut down the circulation of PRRS field virus in this herd. The other thing that I think is important to understand is that in Europe and in North America, we have used Engelbach PRRS MLV for many years as part of either elimination or eradication programs using some of the principles that Dr. Johnson explained in the first presentation. So mass vaccination, herd closure, and just share with you some examples here. 3 cases from Germany, where the predominant strain circulating are PRRS type 1. And in 3 of these papers, the mass vaccination with Engelbach PRRS MLV together with herd closure eliminated PRRS very effectively in those herds. And the 4th publication is a study carried out in North America, where again a parrot-to-finish herd was infected with both type 1 and type 2 strains, and through a program with Engelbach PRRS MLV, the veterinarians were able to stabilize this farm and to start producing PRRS negative pigs at weaning. What about clinical results? Well, here's a few highlights I just want to share with you. One around the performance of the vaccine in relation to pregnant gilts, pregnant sows. What you can see from these 2 publications, both where the challenge was with type 1 viruses. You can see that the vaccine was highly efficacious at reducing the negative impact of PRRS in the breeding herd. And a study a little bit more in detail. This is a controlled study that was done in a breeding herd. And what you can see from this data is that there was significant improvements in reproductive parameters, gestation length, meaning less abortions, the number born alive, but also a lot of positive, significant results in relation to reduced stillborn mummies and mortality rate, and also improved piglet growth from birth to weaning was improved significantly in the Engelbach PRRS MLV vaccinated pigs compared to the controls. And of course, in this herd, the challenge was with a type 1 European strain. I don't want to leave you with the impression that we only have data for type 1. Some of you have probably seen this chart before. We ran out of space to keep updating it, but this is a collection of over 80 Congress abstracts, more than 27 peer-reviewed publications, and more to come since this was last updated, showing across the globe and across time from the last 25 years that Engelbach PRRS MLV shows a very consistent, broad cross protection against emerging strains of PRRS globally. And I would be in trouble with Marius if I didn't share with you that for the new version of this cross protection map, you need to go to PRRS.com. Very soon, you will see this interactive cross protection tool which will allow you to look at different parameters that are relevant to you and get all of the data that shows the benefits of proper PRRS control, including vaccination and all the principles that we've been talking about for many years. So finally, what should your control strategy be, not just for PRRS type 1, but for any PRRS infection? And this is an old message, it's a simple message, like Dr. Johnson says, but it's easy to say, harder to do. And it's 3 key steps. 1, prevent infection. We need to prevent the introduction of new viruses into our herd by optimizing biosecurity, biocontainment, and managing our pig flow to avoid constant introduction of new strains to the herd. Number 2, we need to maximize our immunity. And again, Dr. Johnson's presentation was very clear on how to do that, to stabilize the sow herd and reduce that transmission of sow to pig at the breeding herd level. And this needs vaccination of both sows and piglets. And you need to use a vaccine that gives you the maximum coverage across all of these strains that are out there. And finally, it's really important to minimize exposure. And this is really internal biosecurity. It's managing the level of virus that's in your system to minimize the impact of PRRS. Dose really matters here. So, and when those pigs get exposed. So it is important to manage also the total level of exposure in your herd. So if you remember something about PRRS type 1 or any PRRS, is that we really need to start limiting the introduction of new strains into our herds to successfully stabilize our breeding herds and be successful with our growing pigs. With that, I hand it over to you, Marius.